See, the problem about having an epidural hematoma, which we now know that Ben Chips does, is that it's expansile, it's fast because it's an arterial bleed, and we know that the cranium, the neurocranium, is a closed cavity. And so what happens is that if the middle meningeal artery keeps bleeding, secondary to it being torn from the fracture, then blood's just going to keep accumulating in that epidural space, and because it's a closed container, then there's nowhere for that blood to go. So if we, for example, had a major bleed in the abdomen, then that's obviously not good, but because it's not a closed cavity, that blood will just keep expanding the abdomen. In the cranium, however, something's got to give, and in this case, it's likely to be the cerebrum. And so the reason why he's having motor weakness is that the epidural hematoma is compressing the portion of the cerebrum that is responsible for motor functions. And so we're going to talk about the cerebrum and some of the subcortical structures and a little bit about the brainstem. And hopefully by the end of this, what you'll be able to picture is where the epidural is actually swelling, which part of the brain it's compressing on, and why we're getting those signs and symptoms. So, brain and spinal cord make up the central nervous system. We know this because we studied it way back in week 2, I think it was. So the brain includes the cerebrum, and we've got 2 cerebral hemispheres. Then we've got the diencephalon, which is inferior to the cerebrum, the cerebellum, which is like our little brain, and the brainstem, which you can see here. And we have 3 parts of the brainstem. We'll be discussing them later. Now you can see that this is all brain tissue, and within brain tissue we have grey and white matter. Now grey matter you can see here, and this is referred to as central or subcortical grey matter. And because we're in the central nervous system, we call this grey matter nuclei. And then we have another set of grey matter, and that's peripheral. And we call this the cortex, similar to bone. Okay, as I said, similar to heart, similar to the spleen, lots of things, similar to thymus, lots of things have cortical areas. So this is the cerebral cortex, and these are the subcortical nuclei made up of subcortical grey matter. And then we have white matter. And white matter, which you can see all around here, is going to link the grey areas. Now, some of this white matter is going to be contained within the cerebral hemisphere. Some of this is going to cross over to the other cerebral hemisphere. And some of this white matter is even going to go down to the spinal cord. And we have different names for those types of white matter, and we'll get to that later. So here's the white matter. So here are the 2 cerebral hemispheres. You'll notice that in the center, dividing the 2 cerebral hemispheres, is this longitudinal fissure, sometimes called the great longitudinal fissure. And if you think back to the meninges lecture, then you'll know what sits between the longitudinal fissure, or rather in the longitudinal fissure, between the 2 cerebral hemispheres, and that's the falx cerebri. Remember that sickle-shaped dural reflection? That sits right here in the sagittal plane. But the cerebral hemispheres have to communicate with each other because the left and right hemispheres have slightly different functions, and they're linked by a large bundle of white matter. Now we know we have surface grey matter that we can see here, and that's called the cortex, which we just learned, the cerebral cortex. And that is going to be involved in coordination of different neurons, and it generates its own impulses to control the body. And the cortex that you can see, so this is the great longitudinal fissure that's there. This is, I should point out that this is the white matter there. This is the cerebral cortex, okay? And the cortex has convolutions, and you can see it just here. Now, you might be wondering, why do we have convolutions? Well, the convolutions are there to increase surface area. The more surface area of cerebral cortex we have, the greater number of cell bodies can be placed within that cerebral cortex. Generally, if you're looking at evolutionary biology and comparative neuroanatomy, the number of convolutions that you can see here, you can see all these convolutions. The number of convolutions is proportional to the intelligence of that particular animal, that particular species. So, for example, a few years back, I remember dissecting a sheep brain, and I was struck by the how few convolutions there are in a sheep brain. Likewise, in a horse. I remember dissecting a horse brain, and if that's a reflection on the intelligence, I'm a bit frightened because, quite frankly, a 600, 700 kilo animal with a tiny little brain the size of, almost a fist, scares the hell out of me. Anyway, it's lucky they don't know their own intelligence, otherwise they'll start taking over the world. Their own size, I should say. Otherwise they'll start taking over. And that would be a very frightening prospect indeed. Now, the brain, the only brain that I've seen comparatively that looks very similar to our own is actually the dolphin brain. And in terms of number of convolutions. And so I wonder whether dolphins do have intelligence sort of equal to the human. I don't know. We'll see. Anyway, back to task. So the convolutions are made up of gyri and sulci. Gyri are the hills. So you can see this area here would be a gyrus. And then in between the hills, so here's a hill, here's a hill, so they're both gyri. In between them are sulci. So this is a sulcus here. You can see the sulcus sort of extending down through here. Okay, gyri and sulci. So let's talk about the cerebrum. Here's the cerebrum, and let's start overlaying the different lobes that we have here. So dividing the different lobes are sulcuses. And so all of these different sulcuses, believe it or not, are actually named, but the only ones that I want you to know are the central sulcus here, the lateral sulcus or lateral fissure. If you're using an old textbook, you'll see this referred to as the lateral fissure of Sylvius if we're using the eponymous name, and the parieto-occipital sulcus. Sorry about the spelling area there. Parieto-occipital, so O-C-C-I. Petal sulcus. Then if we overlay the frontal lobe and the parietal lobe on there, what you'll see is that the frontal lobe and parietal lobe are separated by the central sulcus. Now the frontal lobe and temporal lobe here are going to be separated by the lateral sulcus, and the parietal lobe and occipital lobe are separated by the parieto-occipital sulcus. So what you'll see here is that the lobes are named largely by which bones they're most in contact with. So if you're familiar with the skull bones, it's easier to name the lobes. Now we do have another, sorry, that's my drink, I've got some ice in it. Now we do have another lobe, and that's hidden deep to the temporal lobe. So we'd need to remove that, and then we would see what's called the insular lobe, the insular lobe. So let's actually start to go a bit deeper into each one of these lobes, starting with the frontal lobe. We've got three main areas. Prefrontal, premotor is number 2, and just anterior to the central sulcus, so central sulcus is here, we've got the precentral gyrus. Now all contain cell bodies of motor neurons, but most of the motor neurons are located in the area number 1 that you can see here, called the precentral gyrus. So let's have a look at that in detail. The precentral gyrus contains a named cortex. So a gyrus is just this hill, but within a gyrus you can have areas of the cerebral cortex that are defined to a particular function. And so the area, this cortex that's located here on the precentral gyrus is the primary motor cortex. So this controls deliberate conscious movements. So an action potential will be generated here, and this will go all the way down through the white matter, through the brainstem, into the spinal cord to synapse on one of the motor neurons in the anterior or ventral grey horn. Okay, and you'll remember that from week 2. So you can see that if we took a coronal view of the primary motor cortex, you'll see this. And this is basically a sort of diagrammatic representation of what areas of the cortex are actually responsible for control of particular areas of the body. So you'll see in here, so this is going to be where the great longitudinal fissure is, and you can see we have the foot there, the knee, hip, shoulder, wrist, hand, and you can see that the hand's larger proportionally, has a larger amount of cortex devoted to it than, for example, the shoulder. And there's a reason for that. It's because we need fine motor control in the hand. We don't need fine motor control in the shoulder or the elbow because these are gross motor movements, but we do need it in the hand and the face. Okay, face because of facial expression, communication, hand because of manual dexterity. And so this diagrammatic representation of the body on the primary motor cortex is referred to as the homunculus. Now remember that the motor control is contralateral. So the left precentral gyrus containing the left primary motor cortex will actually control movements on the right side of the body. Cool, so here's the motor homunculus. So this is what body would look like if we represented the areas of the body with how many motor neurons are dedicated to that part of the body in the primary motor cortex. So you can see very large hands, very skinny legs, larger feet, but a huge face because we need lots of motor neurons for facial expression. Onward. Here we have the premotor area, and this is a motor association area, and what it's involved in is a motor planning. So what it does is it gets a plan together and then it passes that plan on to the primary motor cortex on the precentral gyrus. So it has different kind of pre-programmed patterns of movement. There's a particularly interesting area here, and it's called Broca's area. And this is dedicated to speech. And this contains motor planning motor neurons that will give motor plans for speech, particularly syntax. And this is an important area because involvement of Broca's area is quite common in certain types of stroke, and that gives us a particular type of speaking difficulty referred to as aphasia. So we get a Broca's aphasia. So if a person presents with stroke-like symptoms with a Broca's aphasia, then it's a pretty good chance that Broca's area will be involved. Cool. The other part, prefrontal area, planning, reasoning, social behaviour, possibly the seat of personality within the brain. The first kind of idea we had about the prefrontal area being responsible for personality came from a man called Phineas Gage. And Phineas Gage was, I apologise, I haven't looked at this kind of story for a little while now, so please excuse me if I'm a bit hazy on the details, but Phineas Gage, from memory, was a railroad worker. And one day, in a horrific accident, he actually drove a railroad spike, so a big old cast iron spike, up through into the neurocranium, straight into the prefrontal cortex that you can see here represented by 3, the prefrontal cortex. And before this injury, Phineas Gage was a very rational, reasonable, pleasant man to deal with. After this, he became very irritable. He made lewd and crude comments, became hypersexualized. He became easily angered, and there's actually there's historical records that his colleagues just said he just wasn't Gage anymore. And that's probably because this railroad spike came through and knocked out his center for personality, the prefrontal cortex. Amazingly, he survived. It really is a true kind of tale for neuroanatomy, and it's kind of the basis for our knowledge in neuroanatomy. And the rest, a lot of neuroanatomy, very sadly, neuroanatomy knowledge actually came through from horrific experiments that they did within concentration camps in World War II, largely in Auschwitz. It's not a fun tale for neuroanatomy. Right, move on. Here's the parietal lobe. Sorry, I should probably make mention just before we move on that the epidural hematoma that Ben has is actually compressing his precentral gyrus. Just to wrap things up, compressing his precentral gyrus, and that's what is causing the motor weakness, because the cells of the primary motor cortex can't generate their action potentials to control skeletal muscles because they're being compressed. Right, let's move on. And let's move on to the parietal lobe. Two main areas. One is the postcentral gyrus that you can see here in the blue. And this contains the primary somatosensory cortex. Big name, primary somatosensory cortex. Also mapped in body areas, and it also receives sensation from the opposite side, so from the contralateral side. And so the left primary somatosensory cortex is housed within the postcentral gyrus, and it receives sensation from the right side. Cool. Number two is the sensory association area, sometimes called the posterior parietal cortex. And so the postcentral gyrus here will only receive somatic sensation, so conscious sensation. But it will be the sensory association area that will actually make sense of what do these particular sensations actually mean? What are they telling me about my environment? If we go posteriorly, we'll see our occipital lobe. Number one that you can see here, this is the primary visual cortex. But this only receives the intensity, color, and shape of a particular object. Intensity, color, and shape. If it wants to make sense of the intensity, color, and shape of an object, it needs to pass that information on to area number two that you can see here. This is the visual association area. So this is going to make sense of all the patterns of light, language, symbols. So when you're reading, you make a hell of a lot of use of your visual association area, represented by two. Temporal lobe. We have an auditory cortex in there. It's important for our sense of smell, and smell also goes to the frontal lobe, and very important for short-term memory and conversion of short-term memory to long-term memory via the hippocampus. Now, deep to the temporal lobe, we have our insular cortex. Important in memory, and it's also important in something that I'm particularly interested in, which is pain perception. And so if you were to try and dissect down to the insula, what you would have to do is you would have to cut along the lateral fissure of Sylvius and remove some of the pia mater that was overlying the gyri and sulci, and you would have to peel it away, as you can see there, and then you can see some more cerebral cortex, and that's the insula. Moving on to white matter. So we've looked at different regions of the cerebral cortex. Now let's look at the white matter. So the two hemispheres, as we know, are separated by the longitudinal fissure, and the longitudinal fissure contains the falx cerebri. But we said before, so here's the longitudinal fissure, but we said before that the left and right cerebral hemispheres have to be connected somehow so they can talk to each other. And the thing that does this is this kind of great, colossal information superhighway. And that is the corpus callosum. So this structure that you can see here and here is the corpus callosum. And there's different regions of the corpus callosum that you'll get into next year. So that's linking the two hemispheres. But what about links between the anterior and posterior portions, for example, of a single hemisphere? Well, for that, we need association fibers. So this is going to link the grey areas within each hemisphere, association fibers. So commissural fibers are going to link between the left and right hemispheres, association fibers between the grey matter in two different places in the same hemisphere. And then we have the third set of fibers, and they're called projection fibers. And these are going to link to the brainstem and spinal cord. All of these are white matter. So let's have a look at some of the commissural fibers in the corpus callosum. So what we've done here is we've actually dissected away some of the frontal, parietal and occipital lobe here, down to the corpus callosum. Again, this is a huge information superhighway between the two cerebral hemispheres. And sometimes, amazingly, as I said before at the start of the lecture, each cerebral hemisphere has different roles, it has different functions. And sometimes and again, very sad, particularly in infants, what will happen is that one cerebral hemisphere is a little bit of a dud, and some infants can experience intractable epilepsy, but it's only in one hemisphere. So in those cases of intractable epilepsy, sometimes what they do is called a cortical hemispherectomy. And so what they do is they actually take an entire hemisphere and the part of the corpus callosum for that hemisphere. And in infants who have this done, they can no longer communicate between the left and right hemispheres. But amazingly, in these infants, due to an incredible process referred to as neuroplasticity, the cerebral hemisphere that's left starts taking over the jobs that was previously performed by the opposite cerebral hemisphere. Absolutely incredible. Right, let's move on. Let's move on to some of the projection fibers now. And the projection fiber, so just to orientate you, this is anterior, this is posterior, this is the cerebral cortex here. And we're talking specifically about projection fibers here. Now, some of the projection fibers from the cortex will sort of be funneled down. So this is all the white matter and it's being funneled down. And this sort of radiating crown of white matter that you can see here is referred to in Latin as the corona radiata, radiating crown. And then this will be these axons, these white matter regions will be funneled down further into what's called the internal capsule. So these projection fibers, these projection axons will be funneled into an internal capsule. So they're encapsulated and they're going to go down towards places like the thalamus, the brainstem, and eventually the spinal cord. So this is all involved in sort of a relay system between different parts of the central nervous system. So let's talk about two main relays. One are the basal ganglia, and really they should be called basal nuclei, but more commonly it's called basal ganglia, but it shouldn't really be called a ganglion, because ganglia, as you know, are collections of grey matter within the peripheral nervous system, whereas a nucleus is a collection of grey matter within the central nervous system. So we should really call it the basal nuclei. And this is the main motor relay. And then we have the diencephalon, which is here. And this is your main sensory relay. Both are contained of grey matter. So these are going to be cell bodies, synapses, and small unmyelinated interneurons. Basal ganglia, two main parts. We have a caudate nucleus, which you can see here. So this whole thing is the caudate nucleus. And so here we have the head of the caudate nucleus, then we have a body of the caudate nucleus, and then we have the tail of the caudate nucleus. And so I kind of think it looks sort of like a little worm, or maybe a spermatozoa that's been curled up. A head, a body, and a tail. Caudate nucleus. Then we have a lentiform nucleus, and that is actually containing two different parts of the grey matter. Now the lateral one that you can see here is the putamen, and then medially this is the globus pallidus. Now the basal ganglia are going to be responsible for controlling motor tone, coordination of motor movements, and it initiates and stops movements. Now if you think about problems with this particular area of grey matter, the basal ganglia, if you think about diseases that affect it, like Huntington's disease, Parkinson's disease, hemibolism, and if you're not familiar with these, then I'm sure you will be over the course of your degree. If you think about these diseases, these are all problems with motor tone, motor movements, and initiating and stopping movements. If you haven't seen any diseases, why don't you just stop this video, do a quick Google on Parkinson's disease, just to get an idea of what happens when this area doesn't work properly. Go for it. I'll wait, I've got all day. Cool. Now, the important thing about the basal ganglia for you guys is to be able to actually identify these different parts on any given sectional level of the brain. Now, I'm only going to expect you to identify these on one particular sectional level. So here we are, and we're about midway down the cerebral hemispheres. And so what we've got is a lot of different stuff, and don't worry about these terms here, but I want you to see the insular cortex. So this area here is the insular cortex. This here is the temporal lobe, part of the temporal lobe, and this division in here is going to be the lateral fissure. And so there's the insular cortex. Now, medial to the insular cortex, you'll see the extreme capsule, which is here, the claustrum here, and the external capsule, and these are all containing association fibers, and I won't test you on these three, even though you may need to know it in future units. I'm not going to test you on those three, but these are the ones that I want you to know. You can see here's the anterior horn of the lateral ventricle, just there, and you can see sort of its bedmate is the head of the caudate nucleus, forming a little impression into the anterior horn of the lateral ventricle, head of the caudate nucleus. Now, just medial to the external capsule, you'll see this is the putamen. Medial to the putamen is the globus pallidus. Medial to the globus pallidus is this kind of boomerang shape, and this is the internal capsule, and you will need to know that. So this is the internal capsule. Right, let's move on to other parts of the central grey matter. Diencephalon, 4 parts: thalamus, epithalamus, hypothalamus, optic chiasma. Let's start off with the thalamus. And here it is. We've got 2 thalami, we've got left and right thalami, and you can see them here. Here's the left thalamus, here's the right thalamus, and the thalamus is made up of many different nuclei. Excuse me. So there we go, there's the right thalamus, there's the left thalamus. And it forms a wall of the third ventricle. So here is the third ventricle in the center here. And it receives info from many sources. Now, the key here is that it's a relayer of sensory information. So what happens is that constantly through your body, there's going to be sensory information transmitted from those sensory nerves through those sensory axons, those pseudo unipolar neurons that we talked about, through the dorsal rootlets, into the spinal cord and up towards the thalamus. And the thalamus sort of has a gatekeeping role to sort of say, yeah, that's important. No, that's not important. Yeah, all right, that's important. So things like the sensation of your clothes on your body, well, that's not really important, is it? So the thalamus says, bugger it. No, I don't want to know about that. If it becomes a problem, let me know. But at the moment, I don't need to send this information up to the primary somatosensory cortex in the postcentral gyrus. Doesn't need to go there. Brain doesn't need to be consciously aware of it at the moment. For some things, like pain, it goes, oh, hang on a minute, could be some damage there. Let's relay that. That's probably important. Right, moving on. Another part of the diencephalon, epithalamus. This little gland here, posterior to the thalamus, is the epithalamus. So this is the thalamus here. This is the epithalamus containing the pineal gland. This will secrete melatonin. I'm sure you've heard of melatonin. Melatonin is a hormone important in your sleep-wake cycles. Responsive to light. So, for example, if you are in contact with a lot of light, then what happens is that the pineal gland will stop secreting melatonin. If you're in a dark space, then it will start secreting melatonin and put you to sleep. So if you want to wake up in the morning, then simply expose yourself to a lot of bright light, suppresses melatonin secretion, and you wake up. Right, another bit. The optic chiasma. You'll see it here. It's where two parts of the optic nerve join. And it's where some of the neurons of the optic nerve split and cross over to ensure they get to their correct hemisphere. Right, diencephalon, hypothalamus. This is what you can see here. It has endocrine and non-endocrine functions. So the non-endocrine we're talking about is sweating, vasodilation, shivering, okay? And these are all autonomic functions. So the hypothalamus is in direct control of the autonomic nervous system. We have another another area, another component, very complex component of our nervous system called the limbic system. This is again in the central nervous system, and we're not going to be talking about it too much because it's extremely complex. All I want you to know about it is it sits largely in the deep areas of the brain, so there are a lot of subcortical nuclei involved in the limbic system, and sits mostly on the medial side of the cerebral hemisphere. Pleasure, anger, affection, memory, and interestingly, olfaction are all going to be processed within the limbic system. And olfaction sort of makes sense that it's going to be sent to the limbic system. Even just from empirical experience, I'm sure that you've almost been kind of taken aback if you smell a certain aroma and suddenly you're transported back to a place or a time or an age, and it suddenly evokes a huge amount of memories. And that's your limbic system at work. Because the olfactory sensations go directly to the limbic system, they are the greatest evoker of memory. Right, let's move on. Cerebellum. We're not going to talk too much about it. It's involved in balance, equilibrium, muscle tone, motor sequencing. Okay? And it has folds of grey matter called the folia, which you can see here, folds of grey matter. And it just sits posterior to the brainstem. You'll remember that between the cerebellum and this part of the brainstem called the pons, we have our fourth ventricle here. Now, internally, in each one of the hemispheres, we have white matter, and this is tree-like, and so we call this the arbor vitae. So vitae is vital, and arbor is Latin for tree. And like nullarbor, treeless plain. Null, no, arbor, tree. And so they refer to the arbor vitae as the sort of tree of life. Now within each cerebellar hemisphere there's two lobes. We have the anterior lobe and the posterior lobe. And these are connected to the brainstem via cerebellar peduncles. I just love the cerebellum. It's there doing its job. It does a fantastic job. It rarely goes wrong, and it's just a fantastic piece of neuroanatomy. It's got almost as many grey matter cells in there as parts of the cerebral cortex have, and I just love it. So the cerebral cortex is always there, always complaining, often going wrong. Cerebellum just sits there and does its job. It's fantastic. It's the workhorse of the brain. Brainstem. 3 parts, and it controls basic functions. This is the oldest part of the brain. If you dissect a rat brain, they have a really well developed brainstem, because these are our kind of our cardiovascular responses, our respiratory responses, control of blood vessels. This is all housed within the brainstem. So we've got 3 parts. We've got the midbrain, and you can see that here in the mid-sagittal, here on the anterior view, so the midbrain would sit here, and here on the posterior lateral view. This whole thing is the midbrain. We've got the pons, you can see that there, again in the mid-sagittal, this whole area, in the anterior view, and on the posterior lateral view here. Finally, the most inferior part of the brainstem is the medulla. You can see that here, here, and here. And what you can see is that we have a whole bunch of different cranial nerves coming out from different parts of the brainstem, which is what we're going to be looking at next. Thank you for joining me.